Congratulations, you made it to the science. I'm Carrie Jennings. I'm a research and policy director at a water nonprofit. Old friends with Ray He. We thought about this project together over coffee in 2020, the week he was closing on the new property. And so I'm going to talk to you about the science team that we assembled to study, from my point of view, the water, but also the air and the soil that goes with it. And it looks like I can change. Which part do I push? There, that one. Partly, the main reason I am looking at regenerative agriculture as a water policy person is because we have a nitrogen problem on our planet. And the watersheds in Minnesota, headwaters of 3 major watersheds, contribute nitrogen to 3 world oceans, and we create a lot of problems with this. Our nitrogen use has increased by more than 10 in the last 4 decades, and that's primarily because we're growing nitrogen-hungry plants like corn and potatoes that require nitrogen but also aren't efficient they don't take up nitrogen efficiently. They lose more than half. And so what we see over the same time frame, you can focus on the little red arrows, nitrogen in our surface water is increasing by over 200% in some of these water bodies. That all leaves Minnesota, so we're polluting the Gulf of Mexico, the North Atlantic, and the Arctic. We know that most of the nitrogen is coming from agricultural sources. Some is produced by fossil fuel production, but primarily it's tile drainage in croplands and then shallow groundwater and surface water. And our major corn growing watersheds are the implicated watersheds. This is a huge financial loss to the farmers in this system. If you look at the average price of nitrogen last year, it's about $1,000 a ton. We're losing $110 million of inputs to these conventional farming systems. It's not just that as a cost, but the state and anybody else trying to drink or use this water has to pay to remove the nitrogen. Minnesota has been put on notice by the Environmental Protection Agency that we have to do better at removing nitrogen from our groundwater, from our aquifers in all of southeastern Minnesota so that it's drinkable for the people there. So this is actually a really good time to focus on the nitrogen problem in the state. But it's not just nitrogen. Nitrogen usually is just an indicator of recent agricultural water that has entered the system, but what goes with that is often scarier. These are data from the county that is immediately north of us, Dakota County. A little bit of Northfield lies in Dakota County. And the dots on the right show you how many other agricultural herbicides or pesticides are found with the nitrogen in the groundwater. And although those numbers are tiny, the red dot is 17 to 25, I think. Different compounds found in the water with the nitrogen. This is an especially vulnerable county with sand at the surface, but this is not unusual for southeastern Minnesota or for much of the agricultural area of the upper Midwest. We know how to protect groundwater, and we know how to stop using as much nitrogen. These are recommendations from Iowa, which is even more in the hands of agriculture than Minnesota. But we know that if we have perennial cover, either through retirement of the land or pasturing of the land, then we don't use nitrogen, and actually those plants will use it up, scavenge it, and keep it from getting into our water system. So this was my entry point into encouraging regenerative practices instead of conventional practices in Minnesota. It's really hard to go up against the corn lobby at the state level or even the federal level. And so what Freshwater thought about doing instead was finding a system of farming that was better for water and supporting that and studying how it improved water quality. So we entered into this project with Ray He on the farm that you just heard described that was purchased in 2020. It happened to be adjacent to an orchard where I knew the owner, and the owner had it under grassland for 30 years. Grass is in between the apples. It's not an organic system, but it has been under perennial cover. So we chose initially to do a comparison of the fields on either side of that electric fence. And all you have to be able to see on this little tackle box full of soil samples is the top is at the top of the box is the top of the soil profile, and then you're going down in columns through different soil series. The right-hand side is the soils that have been under perennial vegetation for more than 30 years, and they should look more crumbly to you. They're going to vary in color, but it's that crumbly texture that turns out to be so important for developing in these regenerative systems, because that texture allows water to infiltrate, nutrients to go into the ground and be held in a way that the roots can then access them. The system on the left is compacted to over 350 pounds per square inch. Water doesn't infiltrate. The tile that were in the system were not even working because water wasn't getting down to the tile. Water was ponding on the top of the fields and that's probably why you got stuck in the mud. And the reason you develop that texture over time is because there's microorganisms that are living in the soil that are kind of gluing together the soil particles in the same way ants do in their anthills. They're creating these little stable aggregates that if you till, you're disrupting. So that's the goal in a regenerative system or a pasture system, is to develop that structure and never touch it. So initially, we went out with John Beck. Many of you met Dr. John Beck last year. He was our state soil scientist. He and I mapped this county together 30 years ago. He is now that he's done with the federal government and our CS position, he's talking about the practices that actually will work. And he and I visited many of the farms in the system to check on which ones we wanted to study long term. But in the process, he explained the soils that were in these farms to the farmers in the system. This last year, I was focused more on policy, and you're going to hear from the other two scientists who are studying, but I brought in some interns just to help me think in particular about how to translate some of our early findings into policy. These were biology students with ecology backgrounds, no soils background, so they took a lot of time this summer to just understand conventional versus regenerative practices, helped Beth install some of her equipment, and then made some policy recommendations after they went to a lot of soil workshops, heard directly from farmers and some other on-farm educators. And these are their recommendations. Their recommendations are that the cost of regenerative practices need to be addressed by policy. There's a relative inflexibility of farming grants and programs available to farmers who want to work regeneratively. And we need more local experts, consultants, and support networks for the farmers that want to try to adopt these practices, and a better dialogue between the state agencies, farmers, and then the scientists studying the systems. We have already started addressing some of these policy initiatives. There's some state legislation. You'll see Beth Fisher on the left there with our representative from Northfield, Christy Purcell. She introduced just this week a bill that favors regenerative practices and supports them in a number of ways. It was introduced very late in the session. It's likely not to go anywhere this year, but it will be carried over for next year, and I expect to see some results then. But this will prioritize soil health in agricultural policy in Minnesota. It will direct some money, especially to the southeastern part of the state where these practices are needed quickly. And we hope to continue to shape existing soil health programs. And I'm going to call out Suzanne Reese, who's in the audience here with the Board of Water and Soil Resources. They have more than $17 million to direct through soil and water conservation districts to people that are working on soil health practices. She has some flyers downstairs and maybe with her right here on the MDA table. So I'm going to turn it over to Beth, and she'll start to talk to you about some of our early results.
Hello, my role in this project is to measure soils, or to measure what's happening in soils. I'm also looking at what's happening in water in the system, working with a team. I have a fleet of undergraduate researchers that I've been working with through the course of this project, and I am very grateful for their contributions. I also am delighted to be working with a fun science team and to be able to work with Ray. Diane wanted us to let you know the reasons we're here. And this list of things I realized in my work as a faculty member, this is my third career, so I'm a little late in the game to get going as a faculty member. And I realized that in my work as a soil scientist, I could take the opportunity to do work that matters for water quality, that matters for climate change, food security, and restoring biodiversity, and equity and justice. And it turns out that regenerative agriculture does all of these things. And so I was pretty delighted to be able to contribute in this way on this project. You've already heard all about this, but I just want to remind you that this is the context that we get to do our work in. The science team has a long list of tasks. Don't worry, I'm not going to talk about all of them in a lot of detail, I promise. I'm going to show you some pictures of the way we're making our measurements, partly because a lot of what we're doing is quite simple. It will, if you have a coffee can, you can do this demonstration to kind of see how it's going on your farm. We're doing it in a way that we're, trying to make it follow a protocol so that we can compare it to other scientific studies. But we're looking at how fast rain can infiltrate into soil in our systems. We're looking at, this one's harder to do at home. Once the rain gets into the soil, what happens? What is the capacity of a soil to store the water? How does it move once it gets in the soil? How fast does it move? So I'll show you the results of these measurements. So as we set up our on-farm kinds of studies, we have to try to isolate a variable that we're measuring. And in this case, we're basically isolating the different land use in contexts. So we tried to keep everything else that we could the same-ish as much as possible. Out in the wild, that's a little bit tricky, but so here's another picture of the latest group. Notice we have a lot of agencies involved. A few of the people there are undergraduate researchers who get to do this work, and I have to tell you that they love it because the mundane, repetitive work that they have to do in the lab has a bigger meaning than just the mundane, repetitive work that we do to do science. I'm telling you, there's a lot of this that is really not that fun and kind of a slog. Okay, so, so one of the places where I'm going to talk to you about today is the tree-range chicken development site. So this site was planted in 2008 and has been under perennial cover for a long time as a result. The contrast site is right across the road. It's been farming corn and soybeans since 1976. They're doing a lot better in their agricultural practices than a lot of the farmers we're seeing in Minnesota. So it's kind of a high bar to compare with, I guess. In their measurements, they're seeing a great improvement in soil carbon. My measurements aren't quite matching theirs, and I think that's because of where you measure it on the landscape. So this is what it looks like when we get to sample. It's kind of a zoo, but it's also really pretty. When I talk about doing agricultural research, people don't realize that we get to be among trees and in grassy areas, and it's quite lovely. This is the contrast site, so it's a really stark contrast from the standpoint of land use and user experience, if you will, while we're sitting out there in the sun making our measurements. So we usually aren't there when the chickens are there, so that's kind of a bummer. So it's kind of novel to get to see them. And Reihi's usually not there either. Anyway, so I have just 2 years of data to show you at 2 different sites. And this is really preliminary. My favorite thing is to have time over several years so we can start to see what patterns emerge. I need you to know that as scientists, our whole thing is learning as we go and modifying our understanding as we learn more. I don't like the current paradigm where everyone is trying to tell you that scientists know all the things. Our whole thing is to be learning. So this is what we're learning right now. One of the things that I measure is organic carbon, and I'm telling you in each of these about the recovery of the soil samples. I guess I should have pointed out one of the students measuring or taking the cores. We do hand coring so that we're a little bit less disruptive. But at Finca Marisol, the development site where tree range was started in 2008, as far as I understand it, it was easy to extract soil on the first year that we did our sampling. And the soil carbon inventory looks great. The place where I think it really shines, I want you to notice that the infiltration is able within the 1st inch, 2nd inch, and 3rd inch to accept 71 inches per hour of rain. So that's a a rate. We're not actually accepting 71 inches. That's like you're driving 55 miles an hour down the road. Okay, so that's the rate. Like, so it's like that. The water's gone, it's infiltrated. So when rain hits that soil, it's gonna stay in that soil. It's not gonna run off. And that's a scenario that we like because water quality. Also because plants need water, I guess. Anyway, so once it's in the soil, it's also capable of moving pretty quickly, which means I think that if we're not in a hill slope, it's going to remain available to plants in the soil. That's what I take that to mean. By contrast, my students get comfortable and very bored when they're on these other sites where they're having to measure 2 inch per hour. That means that it takes a half hour before that inch goes into the ground. Anyway, so they have to, the hardest part is paying attention long enough to take that measurement. So the next year, we had a very dry summer. Maybe you were there for it. It was kind of rough. And our soil probes did not recover as far. So don't even look at that metric tons of carbon per acre. I'm just trying to do an accumulation of it. At some point, we'll normalize them to all at the same depth. So we're comparing fairly across the sites. Oddly enough, the soil was easier to penetrate during this dry phase within the first 25 centimeters. Infiltration, still fast, not quite as ridiculous as it was when the soil was already moist. This is a known soil property. When soil is super dry, it actually is harder to get water in at first. It's kind of like if you have a totally dry sponge, it takes a moment to suck up water in your kitchen. So anyway, we see that in soil as well. I guess physics people know what that's about. Anyway, the water is moving just fine. At Finca Marisol, the contrast, it's harder work to get the water into the soil, and usually the cover on it is bare. So another site that we're seeing that I am able to show you is organic compound. Its contrast site is, again, a corn and soybean rotation, lots of tillage there. What it looks like on the farm is, this is at organic compound. So you're looking at rows of hazelnuts, not down the road, lots of grassy cover. And then the contrast site, we had, it looks like corn emerging at that site at the time of sampling. A little bit more from organic compound because this one has my favorite farmer. All right, anyway, so happy crew there. So organic compound has some of the most ridiculous infiltration rates that we've ever seen. It's amazing. Their contrast site is actually really good infiltration, so there must be something about the general soil properties there that it's able to readily accept rain, and that's a good story for both sites. We see a bit more organic carbon at the organic compound site in the upper horizons, and because the bulk density in this site, the soil is so fluffy, our metric tons of carbon per acre look lower. I'm gonna work on how to legally normalize that a little bit better because yeah, one of the things that we're running into in this site is we seem to not be able to get the coring device down past 60 centimeters, so we're gonna work harder next summer and actually do some separate coring to get it down past that. In 2023, when we again had the dry year, again, crazy, ridiculous infiltration rates that toward the bottom of the graph there. I'm not expecting organic carbon by horizons to change much, but we just didn't recover as deep. And so we don't have as much accumulated here. And again, I should do better about that. So the water is moving in the soil. That's a great scenario. And like I said, we're going to figure out, we're going to keep going, and we're going to dig deeper into the 60 centimeter recovery depth issue. I think it's a parent material story, and we're going to figure out if we actually have carbon storing there and what's happening. Maybe we'll run some water tests in that deeper interval to see if it's a barrier for water and if the storage we see is what's available currently. So promising trends here, we're seeing higher soil organic carbon when we have the regenerative and continuous living cover on the site. That's a good trend. We see really high infiltration where we have the poultry silviculture. We have 5 more years of funding, so I will be able to satisfy my scientific curiosity and hopefully begin to see trends. So you've heard enough about this already, and I'm leaving it there. Thank you to my team. Your turn.
Hi, everyone. I'm Dan Hernandez. I'm a professor here at Carleton College, also part of the science team working on this project. I am going to tell you specifically about the impacts of the chickens on soils in terms of their carbon storage and nutrient cycling. I know that Rehi really likes to say that it's not about the chickens, but this whole talk is about the chickens. So this part, at least, is about the chickens. And spoiler alert, it's really good news. It's really good news for thinking about how this system responds to poultry, even in the context of an agroforestry system. All right, I'm contractually obligated to show this one, right? I have to put this in, but I think we can just move on at this point. When we're studying the process of how these systems change in response to regenerative agriculture, it takes time, right? This is the oldest regenerative poultry site. It's been established for over a decade. We can look at this site and we can see that it's very different than Salvatierra. That's a new site, right? But to really document how these changes happen over time, it takes long-term monitoring, and we're doing that, right? We just acquired 5 years of additional funding to continue this work to take those measurements. But it's also nice to know what changes are happening in a shorter time frame. And so part of what I did with a group of undergraduate students this last summer is think about ways that we could look for patterns for the ways in which these regenerative practices were affecting these systems. That didn't require us to wait 5 years or wait 10 years and see what happens. And so in this study, we really focused on the chickens. And so we just took for granted that this was an agroforestry system, that there were hazelnuts. And then we said, well, there are some places on these farms where there are hazelnuts without chickens, and other places on these farms where they are hazelnuts and chickens. And so we can compare those areas and we can look at how those areas differ. Of course, when we think about understanding, when we think about validating and quantifying the impacts of regenerative agriculture, these pieces that I'm talking about, that Beth and Carrie are talking about, are just one piece of this system. This whole 2 days has been about understanding the impacts on people, the impacts on ecosystems, the impacts on markets, the impacts on the food that we eat, right? So I want to acknowledge that this is one piece. This is one piece that people care about, that investors care about, that consumers care about, and that we should care about in terms of the environmental impacts of producing food. But it's one piece. So the broad question that we're asking in all of this work is, how do regenerative ag practices affect carbon sequestration and soil quality? And I do this work with an amazing group of undergraduates from Carleton that have participated in the work, just like Beth works with students from Mankato State. One of those students is here. Masaki Taniguchi worked on this project last summer. I've now worked with 40 students on this project. Just today, I sent out the emails to accept the 5 that will be working with me this summer. So it's pretty amazing to see these students get involved in this work. And the results of the research that I'm going to show you is part of a paper that we just submitted for publication. The lead author is Maya Hoffman. She's a recent graduate from Carleton, and she's starting graduate school in agroecology in the fall. So it's great. This is having an impact that goes way beyond the science that we're doing, right? Okay, so one thing that we know for sure is that when we put perennial plants on the landscape, things get better. Right? This is a figure looking at time since perennialization and looking at the ways in which going from annual plants to perennial plants, we see improved soil fertility, improved soil carbon, better conservation of nitrogen and phosphorus, and reduced losses of nutrients. This we already know. We know some of this because of work that I've done in the past, looking at the ways in which converting conventional agriculture to restored tallgrass prairie leads to accumulation of carbon in the soil. And this figure over here looks at samples that were taken over a 20-year period and looks at the amount of carbon present in 13 prairies over that time period. So putting perennial vegetation on the landscape leads to more carbon in the soil. We know that that's true. We know that this is true in general. What we don't know is how these patterns play out in this particular system. Right? There isn't a wide literature out there on looking at hazelnuts as an agroforestry model. There's certainly not literature on how that agroforestry model that incorporates poultry impacts these processes. So the question that we're asking is, what about this system? What does this look like? What do we see in terms of carbon sequestration, increases in these benefits to the ecosystem in this particular model? But today, I'm just going to focus on this one question, and that is, what's the chicken effect? How do chickens affect carbon sequestration and nutrient availability in this system? So within this hazelnut system, how do chickens impact that system? This is a cartoon sort of giving you the general layout, but, and this isn't totally accurate. When you go to the farms, you'll realize that the chickens rotate between two areas, but this is showing an area with chickens and an area without chickens. The way that we did this project is that we sampled areas that have had chickens in the past, and we compared it to areas planted in hazelnuts that have never had chickens. Is that what you wanted to know? Is that your question? Yeah, so we looked at places with chickens at any point in the past, and we compared it to areas that have hazelnuts but don't have chickens. Inside the paddocks and outside the paddocks. That's what we're looking at. The hazelnuts are all the same age. In both of the farms where we sampled, the hazelnuts were all planted at the same time. In fact, sometimes you can look down the rows straight through the paddock and outside the paddock, and those are the areas that we sampled. So we're trying to make as close a comparison as we can. We have measured in this system all kinds of things. Beth showed you some of these results. I am going to focus on two responses to the presence or absence of chickens, which is soil organic matter and soil nutrient availability. So all the figures are going to look like this. On the left is samples taken from organic compound. That's Will Crombie's farm back there. And then on the right is samples taken from Salvatierra, which is the new farm. This is the first year it ever had chickens. And then the black bars are the places where chickens were present, and the white bars are places where it never had chickens. Okay? So on the top is soil organic matter, and on the bottom is soil carbon. This is really just two different ways of measuring the amount of carbon in the soil. There's a couple things I want you to see here. One, organic compound has a lot more carbon in the soil than Salvatierra. That's to be expected. Organic compound has been in regenerative agriculture now for, this will be 7 years in this system. Salvatierra is in year 1 when we sampled. So this is a bit of not comparing things exactly because they're different farms in different places, but we can see some evidence that yes, carbon accumulates when you adopt these regenerative practices. If we just compare the areas with hazelnuts, you can see the difference here, right? It's a big difference. It's a 25% difference or something in the amount of carbon in the soil. But we can also see this pattern of what it looks like when chickens are present. We can compare the white bars to the black bars, particularly at organic compound. And there we see, so first, there's more soil organic matter on the older farm. That's cool, that's what we expect. But second, we see that there is a significant increase in soil organic matter and soil carbon, two measures of carbon in the soil that are independent of one another. We see that it is 25% higher in the presence of chickens than without. So Will, you can say for sure that having chickens on that landscape increases the carbon in the soil by 25% compared to having hazelnuts alone for that first 6 years. That's pretty awesome. That's pretty awesome. Yeah, that applause goes to Will and his chickens, right? So chickens increase soil organic matter. And I got a couple new friends here in the front row, and we were talking about poop earlier, and I can't believe I'm going to say poop now twice in a talk in front of all these people. But yeah, chickens poop. And that poop is organic material, and that gets incorporated into the soil, and that builds soil organic matter over time. Sorry, I just had to say poop now 3 times. Okay, what about nitrogen? Well, when we look at total soil nitrogen, we see no difference. The measure of total nitrogen is organic nitrogen and also inorganic nitrogen like ammonium and nitrate. When we just look at inorganic nitrogen, the availability of ammonium and nitrate, we see a big effect. And this is also not surprising, right? Chicken poop has it's high in nitrogen, and so we see the availability of that nitrogen a lot higher in the places where there are chickens. We see this as a significant difference. That's what that little star means at organic compound. It's higher at Salvatierra, but it's not a significant difference. There's a lot of variability on the landscape at Salvatierra. That's one thing that we're seeing a lot at that farm is that it's brand new. It's just getting established. And so the differences are, things are really variable at this point. We also decided to look at this at a finer scale, and we thought about the ways in which chickens impact this system. And in this case, we are comparing samples that we took close to the coop and medium distance from the coop and far from the coop. So that's what you see in these 3 colored stars here, the white star being the samples taken closest to the coop. Our expectation was they go in and out of the coop every day, and so they always have to pass by that spot, and so maybe their impact would be higher in that spot. At organic compound, where the chickens have been present for years now, we don't see much of a difference. We don't see it in soil organic matter. We don't see it in soil carbon. We do see a difference in soil nitrogen. So that's the only one of the 3 where we saw an effect of distance from the coop. But at Salvatierra, chicken impacts are mostly distributed across the paddocks at organic compound. At Salvatierra, we saw that the impacts were much more concentrated near the coops. And so for everything that we measured, we saw this greater intensity of a chicken effect near the coops than far away. Now remember that the overall effect is no difference. It hasn't been long enough. But I think that what this shows is that if you put chickens there for the first time, those effects are going to be more concentrated. But over time, those effects are going to be averaged out over the entire paddock, right? Over 6 years, we start to see those impacts showing up across the entire area. But on the short term, they're going to have the impact on those soils that have really lost soil organic matter, that have really lost nutrients in the places where they're most active. I got just a couple more. We saw a difference for soil organic matter and for soil carbon and for soil nitrogen with distance from the coop at Salvatierra. We also measured greenhouse gas emissions. We use an instrument that we take out in the field, and it measures the emissions of carbon dioxide and methane and nitrous oxide, all 3 greenhouse gases. We saw a few cases where there were individual dates. Those have the little asterisks here. This is looking at the amount of CO2 coming out of the soil taken in weekly measurements at organic compound. We see that there's a few dates where there's a difference, but overall there was no significant difference. When we average across all of the dates, we see no difference in greenhouse gas emissions. So chickens are not increasing greenhouse gas emissions as a result of being in this system. And that's true for carbon dioxide, methane, and nitrous oxide. None of them saw a significant difference. I'm not going to numb your brains showing you all of those figures. I'm just going to show you this one. Okay, so the big take home from this work is that chickens accelerate the soil health benefits of perennialization. Having animals on the landscape, having this integrated plant-animal system accelerates these benefits. That's amazing, because chickens also add some income, right? They're also the thing that the farmers are primarily selling out of the system. And so the benefits, the ecological benefits of this system are enhanced by the presence of chickens. I am just amazed by this finding. It's really cool. And the fact that we were able to see it and see evidence of it established at an older farm and beginning to emerge at a younger farm was really cool. So with that, I will close by saying that being a part of this ecosystem has been a real privilege, right? This is about building relationships. This is about knowing the farmers who we're working with. It's about connecting scientists and students and farmers and all of us involved in this together to try and better understand this system. It's completely different than any type of research I've ever done before this. Most of the time I'm doing work and hoping that someone might be interested in the outcome, right? And in this case, I know because I'm answering questions that the farmers want to know. And that is really gratifying as a scientist to be doing that kind of work. And I think I speak for Beth and for Carrie and for John that we are all just incredibly privileged to be a part of this and really excited to contribute in some way to showing what the impacts are of this kind of system. So with that, we are happy to take questions. We have a few minutes, and I think the two of them can come back up.